Okay, so welcome back to the second video on the fusion pore. Right, so we have seen how we have these core snare complexes which are docking the synaptic vesicle at the presynaptic membrane. Now, when the fusion pore forms, i.e. when the cell receives signal, an action potential in the axon terminal, then what's going to happen is this fusion pore is going to form. Now, one of the hypotheses as to what this fusion pore is that effectively, if we draw the two membranes here, so I'm going to draw them as phospholipid bilayers. So here is the phospholipid bilayer of the synaptic vesicle, okay? Here is the phospholipid bilayer of the plasma membrane, okay? So this is the plasma membrane. These two lines here represent the two layers, the two phospholipid layers that make up the plasma membrane. This is the synaptic vesicle membrane, so synaptic vesicle membrane. Right, so we now somehow need to form a little pore between these, synaptic vesicle membrane. Now, one of the hypotheses is that these pores are made up of the transmembrane components of the snare protein, specifically the snare protein synaptobrevin 2 here, so let's draw those two here, and syntaxin 1 here. Okay, so you wouldn't just have two of these, by the way, you'd have more of them, and they form like a ring. Okay, so in blue is syntaxin 1, in orange is synaptobrevin 2. Okay, and this is the picture if we were looking at it from the side. If we were now to look at it from above, what you would have, i.e. if we looked at it from the synaptic vesicle lumen, what you would have is, if this is the membrane of the synaptic vesicle, you'd have loads of synaptobrevin proteins all sort of forming a ring, basically. So you'd have a ring of synaptobrevin transmembrane domains. And the hypothesis is that this here would form the fusion pore. And then on the plasma membrane, the same thing would happen, but with syntaxin 1 this time. So this would form the tube between the synaptic vesicle and the extracellular fluid. You'd move through this circular array of synaptobrevin 2 proteins, and then through another circular array of syntaxin 1 proteins, and that would form the fusion pore, basically. Okay, so the problem with this theory is that it doesn't really have much scope for lipid being exchanged from the membrane of the synaptic vesicle to the plasma membrane, because we were sort of hoping that this was going to be a lipidic connection, i.e. that the edges of this fusion pore were going to be lipids, so that would sort of give us an understanding of how lipid from the synaptic vesicle could then exchange into the plasma membrane. However, this theory doesn't seem to have any scope for that. However, there is an experiment that you can do that sort of backs this up, which is that you see these transmembrane regions of synaptobrevin 2 and syntaxin 1. So these are these transmembrane regions, often abbreviated to TMRs. Okay? What we could do is we could change some of the amino acids in that portion of the protein, okay, so that they were much bulkier and would sort of stick out into the middle of these circles of the syntaxin 1 protein, and they could manage to block the fusion pore, basically, if we did that. So what we do is we put in tryptophan. We put in the biggest amino acid you can get your hands on. So what we do is we make mutations in syntaxin 1, specifically the transmembrane region of syntaxin 1, and we replace one amino acid in this protein at a time with tryptophan. Now, let me remind you of the structure of the amino acid tryptophan. So, tryptophan then, if we draw the basic amino acid structure first, so here's the amino group, here's the alpha carbon with a hydrogen off it, here's the carboxylic acid group down here, and then the R group of tryptophan is that you then have a methylene group, and then you have what's known as the Indole Ring of this. So let me tell you about what the Indole Ring is. I'll do it over here. But before I can tell you what the Indole Ring is, I need to tell you what the Pyrol Ring is. So we'll start off with what the Pyrol Ring is. So we'll do it in skeletal formulae because it's much prettier if you do it in skeletal formulae. So the Indole Ring then, what you have right at the top is a nitrogen, okay? Then you have 4 carbons, so it makes a 5-membered ring that's a pentagon, like so. So you have 4 carbons, 1, 2, 3, 4, and then this nitrogen up here, and then you have double bonds here and here. So this is a skeletal structure, so these corners are carbons, and wherever there's a free bond, you just assume that's a hydrogen. That's the implicit assumption. So all we have to show then is the hydrogen off this nitrogen. Now, because it often looks ugly to actually put a line there and put a hydrogen, and because I actually haven't got enough space to do that, often you will see people just write the hydrogen in above the nitrogen. So that's a useful trick that might make your pictures look prettier, or if you've got too little space to actually put a bond and then a hydrogen, you will often see people just write the hydrogen above the nitrogen like that. That denotes that the hydrogen is coming off the nitrogen. And all of these 4 carbons, they've clearly all got 1 spare bond, so those will be hydrogens, but because we're talking about skeletal structures, we don't need to show that. Okay, so now, slightly more complicated, the Indole Ring. Okay, so this is the Pyrol Ring. It's an aromatic ring, and this is the Indole Ring next. Now, the Indole Ring is made from the Pyrol Ring. So you take a Pyrol Ring, okay, so let's copy out our Pyrol Ring here. So here's our 5-membered Pyrol Ring. Okay, again, I haven't left enough space to actually put the hydrogen properly on, so I'll just write it above like that. We've got these double bonds here, and then what you do is you attach a 6-membered carbon ring, specifically a benzene ring, onto the Pyrol Ring here. So you have double bond there, and that now is the Indole Ring. So again, these 6 members here are all carbons, and they've all got, well, these 2 no longer have spare bonds, but these 4 here, they all have a spare bond, so that will be implicitly hydrogens coming off there. Right, so tryptophan has an Indole Ring basically stuck off the side of it, and it's basically going to have this whole thing, just move it up there and attach this methylene group onto this carbon here. Okay, so let's do that. Now, usually you actually see the thing flipped over to confuse matters, so, well, confuse the way I've drawn it. So what we're going to do is flip the whole thing over. I hope you can handle this. Okay, and here's the benzene ring off here. Okay, right, so let's put the double bonds in. Double bond there, double bond there, double bond there, double bond there. You need a hydrogen coming off there, hydrogen coming off this nitrogen, hydrogen coming off all of these carbons, and then that is the structure of tryptophan. So basically we've taken that and flipped it upside down, I hope you can see. But we could have just brought it up there and put it in there. It's usually drawn in this way, which is why I've done it that way. Okay, so this is the structure of the amino acid tryptophan, and the single letter amino acid code for tryptophan is W, so you often hear people refer to it as W. Okay, so we're going to mutate certain amino acids in this transmembrane region of syntaxin to tryptophan. Tryptophan then has this big bulky side group that's going to stick out into the fusion pore, and we see whether the conductance of the fusion pore in these neurons with this mutated syntaxin 1 goes down. And indeed, you find it does. So that's an experiment that potentially suggests that yes, the lining of these fusion pores is made up by the transmembrane regions of syntaxin 1. And they did the same thing for synaptic brevin 2 and found a similar result, basically.